NLF, in mostly NLF, non-lactose fermental, the TSI is usually alkaline by alkaline. So this group is again differentiated on the basis of oxidase and nitrate. First we will talk about the oxidase positive and nitrate positive species. In oxidase and nitrate positive, the first one is Shewanella, which is a brown pigment producer, Olegilla, which is non-pigment producer, Elizabeth kingia, which is variable but mostly not produce any pigment, Morguezilla, which is also non-pigmented. First we will discuss the Shewanella. Shewanella is very important organism. The organism produce the brown to tin pigments, and it's usually motile, and as we explained, it is oxidase and nitrate positive, but in additionally it is also positive to the DNAs, and the bacteria is also capable of producing large amount of H2S, and the slant is alkaline by alkaline. This species have two further subgrouping. One is Shewanella petrificans, and one is another is Shewanella algae. Shewanella petrificans required NaCl for the growth, while algae does not require NaCl for their growth. The other Olegilla is also oxidase and nitrate positive, gram negative rod. The key characteristic for this organism is a urea hydrolysis within 5 minutes. The organism is non-pigmented. So this organism is oftenly confused with the Morguezilla olicensis, but Olegilla urelyticus is positive for nitrate reduction and paid, while the Morguezilla olicensis is negative for both of these tests. The another organism in this family is Elizabeth kingia. The organism is very complicated biochemical feature. This organism is also positive to oxidase nitrate and indole, and oxidize the mannitol, ONPG, gelatin hydrolysis, acetylcholine positive. Elizabeth kingia meningospectrum is involved in neonatal meningitis, endocarditis, and nosocomial infection. They are non-pigmented large colonies, and do not grow on McConkey. Another organism in this group is Morguezilla. Is a fastidious organism, only grow on the blood, and the chocolate oxidase test is positive. Morguezilla, for the identification of Morguezilla, we use another test that is called tributrin. Tributrin is positive for Morguezilla species, while negative for Neisseria. These both organisms is gram-negative cocci and been confused during the culture, especially in the sputum culture. The organism have also ability of producing the DNAs, which is a property of Morguezilla catarrhalis, and this is a small plump colonies, gram-negative rod, or sometimes cocobacilli. The organism is catalase positive, also non-motile. DNA is positive but do not grow on the McConkey. Nitrate reduction is positive, indole is negative. In this species, one another subspecies is there, Morguezilla leuconata. It's only proteolytic bacteria in this group. To check this, we use the gelatin hydrolysis 2080 sterase rapid test, and this bacteria also cause pit in the agar in SBA, the greenhouse. Now we will discuss oxidase positive but nitrate negative species. The first one is oxidase positive, nitrate negative, non-motile, non-pigmented. The only thing is the organism is capable of growing on McConkey, and it is also ability of Psychrobacteria petrificans to hydrolyze urea, and a positive test for the paid. Another organism in this group is Rhizobium, which is oxidase positive and nitrate negative, but yellow pigmented and motile. This organism also hydrolyzes the urea, pad, and acetylcholine, which is differentiated from the Psychrobacteria, and also pink colonies, which also aid its identification. It causes the catarrhal sepsis, implant processes, and other bacteria, in the group which is oxidase positive and nitrate negative, but non-motile. It is also pigmented. It's Bacterium, but unlike the Rhizobium is motile, Bacterium is non-motile. The Bacterium have one more identification marker, because this organism also positive for indole, and this organism is thinner at center than end, and they are usually found in the filamentous form. Now we will discuss the weak reaction of oxidase, which is Rosomonas. It is a pigmented pink pigmented organism, can grow on Meconium and SBA. Motility is positive, but weak oxidase positive reaction for Rosomonas species. And Rosomonas hydrolyze the urea and some starches. Another species among this group is oxidase positive, nitrate positive. In this species, myroid species have ability to transfer direct nitrate to nitrogen gas, which is a unique property among all this group. And fruity odor is also a unique property for myroid species. Myroid species was formerly classified as Bacterium, but if you see the Bacterium auditorum is a pigmented yellow colony, can grow on Meconium, but also DNase and gelatin and urea is positive and resistant usually to the polymyxin B. But in myroid case, the fruity odor and direct conversion of nitrate to nitrogen gas is a unique property which distinguish myroid from the Bacterium. Another organism in this group is Spongomonas. Spongomonas is also oxidase positive and pigmented, but motile organism, and its motility is positive in 18 to 22 degrees centigrade, while it is negative at 37 degrees centigrade. Strong yellow pigmented insoluble pigments. Spongomonas species are also able to hydrolyze acetylcholine, but this organism have one unique property, that do not oxidize mannitol. Another species which is quite similar to the Spongomonas but non-motile, and it can grow on SBA, and that is Spongobacteria. Spongobacteria is a gram-negative rod, yellow pigmented colony, non-motile, and oxidase positive, nitrate negative. This is further two species which is clinically important. One is Spongobacteria multivorum. It's found in variety of clinical samples, and oxidized glucose and xylose, while the Spiritorum is recovered from the blood and urine and only oxidize the mannitol. Now we will discuss the oxidase negative and nitrate negative and two species. One is Acinetobacter and another Methylobacterium. Acinetobacter baumannii is oxidizing, its have ability to oxidize the glucose, while Acinetobacter lufi and Acinetobacter hemolyticus do not oxidize the glucose. The Acinetobacter baumannii is non-hemolytic, while only species is Acinetobacter hemolyticum that show the hemolysis. So this the another species in this group that is oxidase negative and nitrate negative is the Methylobacterium, that is gram-negative rod, large vacuolated with volatile granules causing some nosocomial infection like septicemia and perinatal dialysis. The organism is capable of producing the coral pink colonies on SBA. Motility is positive, and oxidase nitrate is negative. Indole is also positive. The organism use the methanol as a sole carbon source for its biochemical processes. Methylobacterium species also hydrolyze the urea and some starches. This is another chart for gram-negative non-lactose fermenters. And again, this is classified on the basis of oxidase and nitrate positive organism, especially in the cystic fibrosis patient, or you can say the Burkholderia is very important. Burkholderia is encapsulated bacterium, and they have further 4 species. And the first one is lysine positive Burkholderia capsica, which is motile, and Burkholderia pseudomyli that cause the melioidosis but positive for lysine. Among this group, the yellow pigmented bacteria, which is a unique property for this species of Burkholderia, is Burkholderia godolai, and this is oxidase negative. Burkholderia milli is a known cause of glandular disease, but it is mannitol positive, which is a unique property to distinguish it from all other species. Dilithia acidovirens cause the eye and ear infection, and mannitol positive. Spongomonas testosterone in cystic fibrosis infection is a pigmented tints to brown pigment and mannitol negative. And the media for this whole group is oxidation fermentation polymyxin B lactose media, which is used for this group to check the different oxidative property of all the bacteria, because this group have only oxidative properties. They do not ferment glucose. Rosomonas picti is a biofilm producer pseudobacteria causing the nosocomial outbreak. Bacteremia, endocarditis, meningitis have ability to oxidize glucose and xylose only. Rosomonas insidosa is only xylose positive, while Rosomonas mannitolitica is mannitol positive and also oxidized xylose, lactose and maltose. Among this group, the nitrate negative group, which is oxidase positive but nitrate negative, are Caperoviridius species and Burwadomonas. Burwadomonas is further divided into species Burwadomonas diaminota. They do not ferment the maltose, and Burwadomonas vesiculoris, which ferment the maltose. Among this group, which is oxidase negative, nitrate negative, is Stenotrophomonas maltophilia, is a motile and DNase positive organism.